Computation and System Biology Assignment Help. For any assignment-related queries, call us at +1 678 648 4277. You can mail us at info@nursingdassignmenthelp.com or reach us at https://www.nursingassignmenthelp.com. To better understand inborn disorders of metabolism, you isolate a strain of mice that becomes ill unless fed a diet lacking phenylalanine. You sequence the genome of this mouse and find several differences from wild type, including a change to a region that encodes a highly expressed 68 nucleotide RNA, which has sequence 5' ugwakogagagukogkakigog ACUGCACGUCACCCUCCUGAI3 in your strain and 5 ugwakogagakagukukukukugagaya ACUGCACGUCCCUCCUGI3 in wild type mice. Search the sequence in your strain against the mouse genome and transcriptome. Computation and System Biology Assignment Help. Search the sequence in your strain against the mouse genome and transcriptome using NCBI's BLASTN. From the BLAST homepage, click on Nucleotide BLAST, not mouse, and use the Mouse Genomic Plus Transcript G+T database, optimized for somewhat similar sequences. By expanding the algorithm parameters box at the bottom, set the match/mismatch scores to +1/-3. A. How many statistically significant hits are there at an e-value of 0.05? In one sentence, what does an e-value of 0.05 mean? For transcript hits, what are the maximum reported scores, and are they raw scores or bit scores? Click on the hyperlink to view individual hits. To what parts of your RNA do these hits correspond, and what is the percent match? Nursingassignmenthelp.com. B. Using the e-value and reported score from the result with the highest percent identity match from part A, calculate the approximate length of the mouse G+T database. Using the score S equals 50.1 bits and e-value equals 210, for along with M equals 68 and T in the formula E minus value equals 2. S yields a mouse G+T database length of equals 3.55109. Note that the mouse haploid genome assembly is about 2.7 billion base pairs, so after adding in, there are 2 transcript and 2 genome hits at an e-value of 0.05. The e-value is the expected number of hits with score at least as high as the hits reported score when searching a query of length 68 and T against the mouse G+T database. The maximum scores for the 2 transcript hits are 54 and 50.1 bits. The hit with score 54 bits corresponds to positions 38 to 68 of the query and has 97% identity to its match, matches 30 of 31 positions, while the hit with score 50.1 bits corresponds to positions 14 to 38 of the query and has 100% identity. Mouse haploid genome assembly is about 2.7 billion base pairs, so after adding in transcript sequences, the estimate from the formula is around what we would expect. Various corrections to the simple formula are made for base content, repetitive regions, and other parameters for the reported BLAST values. C. Consider a query sequence Q of length L that matches perfectly to a sequence in the database, yielding a BLAST e-value E1. How would the e-value change if only the first half of Q were searched against the database? In particular, would it stay the same, go up, go down, and how, linearly, exponentially, etc.? E1 equals 2, S and E2 equals 2. S/2E12S equals 2E22S/2E2 equals E12S/2. 1. Thus, the e-value increases essentially exponentially, with an additional decreasing linear factor of 2 due to having M. But this latter effect is much smaller than the exponential increase resulting from the decreased score. Nursingassignmenthelp.com. Intuitively, decreasing the length of query, and therefore match, should make the match more likely simply by chance and therefore less significant, so we should expect the e-value to increase. Quantitatively, if the sequence query length were halved, /2, the score S would decrease by a factor of 2, SS/2, since there are half as many positions at which to accumulate positive match scores. Plugging these into equations for the original query sequence, with score E1, and the half-length query sequence, with score E2, yields D. Returning to the BLAST results from Part A, to what genes and RNA classes do the transcript hits with E-values below 0.05 belong? Does your RNA match the sense or antisense direction of these hits? Click on the hyperlink of the hit and look at the strand section, which tells you the DNA strand of the hit/query. Of the two statistically transcript significant hits at an E-value of 0.05, one matches nucleotides 1438 of your RNA complementary to matching the antisense direction of an mRNA that encodes the phenylalanine hydroxylase PAH enzyme. Nucleotides 38 to 68 of your RNA match the sense direction of SNORD 100, AC/D box SNO RNA, a type of non-coding RNA that directs post-transcriptional modifications of other RNAs. After performing an RNA protein affinity purification, pull down from mouse cell lysates followed by mass spectrometry, you determine that your RNA interacts with the product of the ADR1 gene. What does this enzyme do, and what type of RNA does this enzyme act on? Looking back at the function and strand of the gene hit to the second part of your RNA, state a hypothesis as to how your RNA might function to cause your mouse's metabolic disorder. Hint: on the BLAST hit entry corresponding to the mRNA, click on the graphics link to see the hit in red and how your query at the bottom overlaps with it. If ADAR1 acts at the UAU codon, what is the resulting change during translation? NursingAssignmentHelp.com. The ADAR1 enzyme catalyzes A2I editing, post-transcriptionally deaminating adenosine in double-stranded RNA duplexes, yielding inosine. Since I is interpreted as G during translation, A2I changes in protein coding sequences may lead to codon changes and altered functional properties of the proteins. In addition, A2I editing can play important roles in regulating gene expression, such as by altering alternative splicing, miRNA sequences, or miRNA target sites in the mRNA. The PAH gene product is a critical enzyme in phenylalanine metabolism and catalyzes the rate-limiting step in its complete catabolism. Nucleotides 14 to 38 of your RNA overlap a region of the PAH-ORF antisense to the mRNA, including tyrosine 414 encoded by the codon UAU. Deamination of this adenosine by ADR would result in the ribosome interpreting a UGU codon, which encodes for the much smaller cysteine. Thus, your mutant SNO RNA provides an RNA duplex for ADR1 to cause a missense mutation, which could resulting in reduced activity of the PAH enzyme and contribute to your mouse's metabolic disorder. Indeed, genetic Y414C mutations have been observed in human phenylketonuria patients, and the mutation has been shown to induce global PAH conformational changes, Gersting et al. AM Journ. Human Genetics 83, 2008 http://www.ncbi.nlm.nih.gov/pmc/articles/pmc2443833/pdf/main.pdf. In order to perform this alignment, you must first choose a scoring matrix. For example, you could use a constant match and mismatch penalty of 1 and -1, respectively, so that SIG equals 1 if I equals J and SIG equals -1 otherwise. Is this a good idea? Why or why not? In one sentence, briefly describe how you might obtain a better scoring matrix for protein comparison. No, not all amino acid substitutions are equally